Okay, very good morning, my dear friends. Today's our eminent and very experienced and very energetic and social service oriented personality in front of us. So, in the regarding of CPGET 2023, CPGET 23 PG entrance online coaching, free coaching. Last year also, Dr.M.Rambabu sir has served for so many classes. His huge experience is very useful and they get benefited and join in various universities. So many friends. So my dear friends, I would like to invite today's our eminent resource person, Dr. M. Rambabu Garu, Assistant Professor, Department of Botany, NSS Program Coordinator, and MISAL, NSS MISAL Oasis, Telangana State Regional Coordinator, Dr. M. Rambabu Garu, Kakatiya Government College, Warangal. So please welcome, sir, and share your screen and start your session. Before starting the session, please speak few words regarding of this program and your experience in previous last year also. Welcome.
Thank you, Dr. Tirupati sir, for giving me this opportunity. Sir, are you audible? Am I audible?
Yes, you are audible, sir.
Yes. So thank you once again, Dr. Padala Tirupati sir, Assistant Professor of Botany, Secretarial College. Thank you for giving me this opportunity. So I'm very happy to share my views regarding this free coaching online CPGET coaching from last 2 years, I think. So in previous year also, we have given a lot of services to the students and as well as the beneficiaries. No doubt about that. 100% it is very, very valuable and very useful program to the upcoming PG entrance holders. Once again, my heartfelt congratulations to you as the organizer to conducting these kind of valuable program. The previous year also, we got from this entrance we got almost all 4 best ranks. Below 100, 4 ranks we got in a different university entrance level. So this year also, I'm expecting the same 100%. The beneficiaries, I expecting the same ranks in upcoming entrance examination also. My heartful wishes and the heartful blessings to the students for their upcoming entrance examinations. Thank you for giving me this opportunity. I'm ready to take the class now. So I'm going to take the class on plant tissue culture. So plant tissue culture is a wonderful topic. I think 2 or 3 questions may come in your entrance examination from this topic. I want to share my views. Some technical problem is there. I think. Please wait. Okay, I will share my screen. So visible, sir? Tirupati, sir? Tirupati, sir?
Yes, sir, it's visible.
Visible. Sorry for technical problem. Now, plant tissue culture. So actually, this is very, very important topic in our entrance and as well as upcoming theory examination also. So coming to the definition part, what is actually plant tissue culture? The definition is the culture of plant, seeds, organs, tissues, cells, or protoplasts, and nutrient media under sterile condition. This is the perfect definition. Whatever it may be, the culturing of plant materials, the culturing of plant materials on suitable nutrient media, and then production of the plants process is called as tissue culture. Simply, tissue culture. Culture is nothing but a production. Tissue culture. We can produce our plants by the using of the tissue. That is a simple definition of tissue culture. You see, agriculture, aquaculture, sericulture. Agriculture means production of the crops, culture, agriculture. Similarly, tissue culture. We are using the tissues for the production of plant crops. That is the meaning of tissue culture simply. Got it my point, Al?
Yes, sir.
Right. So coming to the, what is the actual background? Tissue culture, that tissue culture process has begun at the early stage of the 20th century. Of course, he's a great man. You see, Gottlieb Haberland, scientist. He's from Germany. He is considered as a father of tissue culture. Why? Because this is important question, my dear. Note down. Father of tissue culture is Gottlieb Haberland. Who is the father of tissue culture? Haberland. So this is the entrance question. So he, first of all, he produced the complete plant by the using of the cork tissue. He inoculated the cork tissue on the nutrient media. By that, he has produced the plant. That's why the first credit goes to the great scientist Haberland. That's why this Haberland scientist is considered as a father of tissue culture. So these are some more important historical, issues in our tissue culture. One or two questions may come in your examinations. These are the cell theory, Schleiden and Schwann. We know that. Okay. Then first attempt of tissue culture, Haberland, continuously growth callus culture, White. Then like this, these are shoot tip culture, Ball, and organs culture, regeneration, Ball. So plant from single culture, more. Then protoplast isolation causing. This is important, Nana. Last one, you see, in 1960, protoplast isolation, first protoplast isolation developed by the Hawking scientists. This is important. Last one, my dear, once again I am repeating. Protoplast isolation technique first developed by scientist Hawking. Important. Like this, MS media. The media type of the media MS, that is Morashige and this Cook. Like this, some important aspects are there. I will send this PPT to your group also. Just you see once again after completion of our class. Then coming to what are the actual basis of the plant tissue culture? What are the major requirements? Major requirements, they are the plant growth regulators, plant growth hormones. These are very, very important. They are auxins and cytokinins. Auxins and cytokinins are plant growth regulators or phytohormones. These auxins, auxins are generally used for the stimulates the root development. Cytokinin stimulates the shoot development. This is common. Auxins are used for root development. Cytokinins are shoot developments. But the quantity is also important. How much auxins are useful? How much cytokinins are useful? Similarly, you see, high auxin, low cytokinin. First one, high auxin, low cytokinin. Results, root development. Then high cytokinin, low auxin. Results, shoot development. The third one, auxins and cytokinin in equal concentration. Auxin and cytokinins are equal concentration. That is causes for the callus development as well as regeneration. High auxins, root development. High cytokinin, shoot development. Both are same auxins and cytokinin, same in quantity. That is helpful for the callus induction as well as regeneration or organogenesis. Regeneration means production, reproduction. That is regeneration. Organogenesis means organs generation. So these are the needed. What are the basic needs? First of all, you see, appropriate tissue. No, for the purpose of tissue culture, we must be take the healthy and young tissues. We must be take the tissues in healthy condition, not in diseased tissue. If you take the diseased tissue, the total experiment will be failed. That's why we should take the healthy and young tissues for the purpose of the tissue culture process. That's the first thing. The second thing is suitable growth media. You know, generally we are taking that MS media, MS medium, that is called as Murashige and Scoop medium. That MS medium is common type of medium for the process of tissue culture. Some more types of mediums are also there, you know, like WPM medium or, you know, B5 medium. Different kinds of medias are there, but in general, the suitable medium is MS medium, that is Murshik and Scoop medium. That is important. Then aseptic condition, third one. The entire experiment is always under aseptic condition. The entire experiment always under aseptic condition. Why? Because the media is artificial. We are growing the tissue on artificial media. So number of microbes will be attacked. That's why the entire experiment carry always under aseptic condition, sterile condition. That is important. Then fourth one is growth regulator. Already we have discussed auxins, cytokinins, gibberellins, etc. These are important. Then environment factors, you know, light, temperature, related humidity and sterile condition. In general, plants grows in normal condition with the suitable light intensity. For the purpose of growing, for the purpose of photosynthesis, the suitable light is required. That much light we should arrange in the tissue culture room. In normal condition, sunlight is there. That is used for the photosynthesis and survival to the plant. That much light intensity we should arrange, we should create in the laboratory of the tissue culture. That is important. Now, temperature is also, room temperature must be maintained in the tissue culture, 25 plus or minus 2 degrees centigrade. That is the 25 plus or minus 2 degrees centigrade. That is the suitable, you know, quantity, intensity of the temperature. Then sixth one, frequently subculture. After the culture, after 30 days, that material will be transferred to the fresh media. That's called as subculture. After the culture, whatever it may be, regenerated, you know, callus or organogens will be developed on the nutrient media. Then after 1 month, those material will be shifted to another fresh media. That is culture, that is subculture. So these kind of 5 important needfuls are very, very important. Appropriate tissue, that is healthy tissue, suitable growth media, MS media, sterile condition, then growth regulators like auxins, cytokinins, gibberellin, environmental factors like light, temperature, no humidity, then finally frequently change that material from one media to another medium. So what is actually tissue culture process? Is the process of growing cells artificially in the laboratory. Generally, the tissues are developed, maintained in the laboratory. That is, this entire tissue culture process is a artificial process. We know that we are culturing the tissue on suitable nutrient media. That is artificial process maintained in the laboratory. Then based on cellular totipotency, this is important terminology, totipotency. What is the meaning of totipotency? You know that. Each and every cell of the tissue explant, the used explant, each and every cell have the capacity of production of new plants. Whatever it may be callus, whatever it may be explant. In that explant, each and every cell have the capacity of production of new plant, which is similar to its parent plant. Each and every cell produces that new plant, which is similar to its parent. So that is called as cellular totipotency. That cell has the potential level, which is produce the new plant similar to its parent plant. That is totipotency. Now this is next one. This is in vitro technology. You see, in vitro means inside of the laboratory, in the laboratory, in the test tube. This technique is in the laboratory and in the test tube. That's called as in vitro. In vitro means inside of the test tube and inside of the laboratory. And next one, true to type plant production. You see the terminology, true to type. The tissue culture produced plant. The tissue culture produced plant almost all similar to its parent plant. The tissue culture generated new plant is similar to its parent plant. That term is true to type plant. And then lab to land program. This process is a lab to land program. Generally, tissue culture process developed in the laboratory. The plants are germinated from the tissues in the laboratory. And from that, the plants will be transferred from the lab to outside environment, that is land. This process is lab to land program. Inside of the laboratory, we will develop the plant. The new developed tissue cultured plants in the laboratory are shifted to land, outside field. That is lab to land program. Then finally, mass multiplication technology are also called as micropropagation. In general, whenever we are going to sow a seed in the soil, for example, you should imagine, whenever we are going to sow a seed in the soil, what happens automatically after some days? That seed will be germinates to plant a single plant. Germinates to a single plant. And each and every seed is responsible for a single plant generation. That's common. But here in the tissue culture, we are inoculating a small explant in the laboratory. That small explant can produce a number of plants. A seed will respond, a seed is germinates to form a single plant. Seed is germinates to form, seed is germinates to, a single plant that is responsible in normal condition. But here, a small explant can regenerate to produce number of plants. That is called as micropropagation. Small tissue can produce a number of plants, also called as mass multiplication. So these are the very, very important aspects. This is completely artificially programmed in the laboratory based on cellular totipotency. This is in vitro technology, true to type technology, lab to land program, and mass multiplication or micropropagation. So these are the very, very important aspects in the tissue culture. This is the introductory part of the tissue culture. Very, very important. These are the types of cultures. Which type of explant is going to culture? Seed culture, embryo culture, epidiole culture, leaf culture, stem culture, root culture, nodal culture, shoot-tip culture, anther culture, ovary culture, and protoplast culture, etc. Which type of explant we are going to use, that is the type of culture. Simply, this is the black diagram of the tissue culture. We are taking the explant left side, you see, leaf, node, you see, leaf, node, shoot-tip, anther, ovule, seed. So these are the explant left side. Those will be cultured on the nutrient media that is called as inoculation. After some days, these, explant is converted into callus called as callusing. By the effect of auxins and cytokinins, these explant will observe the nutrients from the media. This explant will observe nutrients from the media, and also by the stimulation of auxins and cytokinins, this will be converted into callus. The production of callus is called as callogenesis or callusing. This callus will be shifted and again shifted to cytokinins media. By the effect of cytokinins, this callus produces the plantlet that's called as regeneration. So these plantlets will be shifted to rooting media. Means that media have the auxins. With a suitable type of auxins, the plantlets produces the roots. These were shifted to the greenhouse that is called as hardening and acclimatization. Finally, these plants are shifted to ground. So completely, this is the black diagram of the tissue culture. Steps. This is your upcoming examination. What is tissue culture? Write the steps which are used in the plant tissue culture. That is a question or coming to your entrance examination also. Steps. What are the steps in tissue culture? First one is nutrient media preparation, sterilization, that media, explant preparation, inoculation, incubation, hardening and acclimatization. These are the 6 steps which are involved in plant tissue culture. We are going to see one by one. This is the nutrient media preparation. Take a distilled water and add a suitable quantity of the following. First of all, in the laboratory, you take the distilled water in the suitable quantity of the conical flask or culture flask. Then add the following inorganic nutrients, organic nutrients. So in the inorganic, micronutrients are there. In organic form, carbohydrates, vitamins, amino acids, like materials are there. And we see add plant growth regulators also. What are the plant growth regulators? Auxins, gibberellins, cytokinins. So these only the growth promoters. Auxins, gibberellins, cytokinins only, not abscisic acid. Auxins, gibberellins, cytokinins. These 3 are only growth promoters. And then after adding these, organic and inorganic substance and also plant growth regulators, you take the pH of that particular media. Must be measure the pH value. That is very, very important. pH value always in between 5.6 to 5.8. Note down, my dears. In the plant tissue culture, the pH value is 5.6 to 5.8, approximately 5.7. This is the perfect pH value for the purpose of tissue culture process. The media pH value is 5.6 to 5.8, around 5.7. Then finally add agar-agar. So agar-agar is a wheat powder like substance which is used for the semi-solidify. The liquid form is converted into semi-solidify by the adding of agar. Then coming to the sterilization of nutrient media. After the preparation of that nutrient media, those media must be sterilized. Otherwise, number of microbes will be attacked. That's why the prepared nutrients media must be sterilized with the help of autoclave. You see, this is the autoclave. Already you have seen in your college. This is the autoclave. This autoclave, working is based on the pressure cooker. Similarly, pressure cooker. The working is similarly pressure cooker. In that autoclave, we put the cultured vessels or cultured bottles in the autoclave, then switch on. This is worked with the help of electricity. So in that autoclave, the measurements are 121 degrees centigrade temperature, 15 pounds of pressure. In that 121 degrees centigrade, 15 pounds of pressure, the time is 15 minutes. Culture bottles, culture wells put in the autoclave, then switch on. When the temperature is accurately reaches to 121 degrees centigrade, the inner side pressure is also reaches the 15 pounds. In the two quantity of the measurement, 15 minutes time you should take. In that condition, the entire media will be sterilized. That is the second step, nutrient media sterilization with the help of autoclave. Then the third one, explant preparation. Why? What is the explant preparation? Explant is already present in the plant. We should take the explant like leaves, stem, or shoots, nodes, internodes, different kinds of the plant parts are there. How can we prepare? The preparation means here is prepare for the inoculation. The plant parts are ready-made is there. The plant parts are there, but directly we are not going to put that parts on the nutrient media. First of all, you take that plant parts and prepare for the inoculation. That is the explant preparation. Take the, for example, you take the leaf. Take that plant part of the leaf and wash with running tap water. That's called as rinsing. Wash with running tap water. Then going to wash with liquid detergents for 3 types. Liquid detergents. Then after wash with fungicides. Fungicides. They are mercury chloride or sodium hypochlorite solution. With the help of these fungicides, see what are the fungicides? 0.1% mercury chloride or 12 to 13% of sodium hypochlorite solution. The plant pots are always full of microbes. Why? Because they are open. The plants are always opened in the atmosphere, natural atmosphere. By that only, number of microbes will be affects the plant pots. So by the removing, for the purpose of removing of these microbes, we must be washed with fungicide. By the effect of washing of these fungicides, the microbes will be removed from the plant pots. Then finally wash with sterile distilled water. Then that parts will be cut into small pieces. Finally, wash with sterile distilled water. Then cut into small pieces. The small pieces size is almost 3 to 5 millimeter size. That 3 to 5 washed plant material is called now explant. We have taken the parts, washed with different levels, and cut into small pieces. The cutted small piece is now called as explant. It is ready to, inoculation. That is the process of preparation of explant. This is also block diagram, the plant tissue culture, which is present in your books also. Then the fourth step is inoculation. Inoculation means the prepared explant. Already we have discussed. the prepared explant is placed on sterile nutrient media. In the first step, we have prepared a nutrient media. In the second step, that media sterilized by the autoclave. In the third step, explant prepared. Now what we are going to do, that prepared explant, the sterilized explant is placed on sterilized nutrient media. That process is called as inoculation. This inoculation process is always under aseptic also. Media sterilized, explant sterilized, inoculation is also under aseptic condition. For the purpose of inoculation, nowadays we are using inoculation chambers. Inoculation chambers. That inoculation chambers names are laminar airflow. Laminar airflow. That chamber name. See here, you see that scientist is going to inoculating the different material and suitable nutrient. This is the inoculation chamber. A big size chamber is inoculation chamber, laminar airflow. In the inoculation chamber, the sterilized explant is going to placed on sterilized nutrient media. That process is called as inoculation. For the purpose of inoculation, we are going to take the inoculation chambers. They are now called as laminar airflows. Got it? Then, the fifth step is incubation. After completion of inoculation, that inoculated tissue cultured, inoculated test tubes or culture tubes. After the inoculation, those tubes are called as culture tubes or culture bodies. Where we should be kept them? So those cultured bodies or cultured tubes will keep on a suitable rooms. Here you see, these are culture rooms, incubation rooms. After the inoculation, those will be, this is the tissue culture incubation room. The culture bodies. After the inoculation, these culture bottles are shifted to incubation room. You see, this is the incubation chamber, like a bookshelf in the library. This incubation room always under sterile. This is also always under sterile. Media sterile, explant sterile, inoculation sterile, and incubation is also sterile. Always under sterile. Here the temperature is maintained around 25 plus or minus 2 degrees centigrade here. You see, the light intensity approximately 5,000. Here you see, around 5,000 lux intensity of light we should supply. The temperature is 25 plus or minus 2 degrees centigrade. The related humidity inside of the humidity maintained 50 to 60%. The photoperiodism, the light time 16 hours. You see, my dear, here the temperature and humidity and light intensity, the light intensity time, all these are artificially we are providing to the tissues in the incubation room. But in the natural condition, all these are maintained in outer atmosphere. So like that, outer atmospheric conditions we are creating in the laboratory. So these are very, very important measurements. Time of light intensity, quantity of the light intensity, humidity, and the temperature. Always maintained in this, you see, incubation room. After what happens after 1 month, this inoculated explant will be converted to callus. Inoculated material converted into callus. By the effect that callus will be transferred. Already I have shown the background, the black diagram before before discussing these steps, we're inoculating the explant on nutrient media after some days. That explant, convert into callus. Those callus will be shifted on another cytokinin media. That's why the organs will be developed called as organogenesis. Then organs will be, shoot tips. The shoot tips will be cut and shift to the rooting media. What happens? That roots will be developed. The cut end regions of the, fresh and test tube plantlet produces the lateral roots. The cut end region part have the pericycle. You know the function of pericycle. The pericycle cells are used for the lateral roots. The pericycle region cells produces the lateral roots by the absorbing of oxygen. That's why roots will be developed. That's called as rhizogenesis. So all these developments are seen in this incubation room. What happens actually after the completion of this development? These plants will be shifted to transfer to outer environment. That's called as hardening and acclimatization. Transfer the plantlets from culture media to outer environment. This is very, very important, as called as lab to land program. Here you see, this is the hardening and acclimatization. In previous 5 steps in the laboratory, the 5th step is incubation. In the incubation period, the plants have developed. The developed plants will be shifted to the outer field. Those will be shifted to the outer field. This is called as hardening and acclimatization. This is very, very important step for the purpose of surviving of these test tube plants. So this is a very, very important step. These are all steps. Very, very important in the plant tissue culture process. Once again, you see hardening and acclimatization, right? This is the incubation chambers. This is the inoculation process in the lab. This is autoclave for the purpose of media sterilization. I have shown this black diagram. So these are the egg plants cultured on nutrient media. Then after callus production, then after callus regeneration, then plants will be, the cut end regions of the shoot bud developed the roots. The complete plant developed. These complete plants will be shifted to the outer environment. That's called as hardening and acclimatization. So this is the lab to land program. So this is the my part, that is
Okay, my dear friends, regarding of today's class, our eminent botanist, Dr.M.Rambabu sir has clearly explained. Sir, what about you? What about your experience? What about if you have any doubts, Mr. Prakash?
No, sir. No, sir.
Any doubt regarding of this class? Please you ask our respected resource person.
No doubts about.
Okay, speak, my dear friends. Please ask any doubts. Prakash,
No doubts, sir.
Any other participants? So really, it is a wonderful class, wonderful presentation. Sir has clearly explained about the entire process of the plant tissue culture in a single class, but the single class contains the entire tissue culture techniques and tools of the plant tissue culture and method of the plant tissue culture. Even though the beautiful inoculation chamber, as sir has shown about the how to inoculation process in the laminar airflow chamber and also hardening. So each and every step sir has clearly stated in this class. Am I right, my dear friends? Mr. Shivaji Naik.
Yes, sir. Yes, sir.
Okay, Shivaji Naik, Rambabu sir.
Yes.
Shivaji Naik is our previous student. He has got first rank in Andhra Pradesh state among 11 universities. He has got in his reservation. He has got first rank in Andhra Pradesh state CPGET 2021.
Oh my God.
Am I right,
Hearty
Prakash?
congratulations.
Am I right,
Hearty
Shivaji?
congratulations.
Yes, sir.
Shivaji,
Yes, sir.
hearty
Thank you, sir.
congratulations.
Thank you, sir.
So,
God bless you.
in this
Thank
regard,
you, sir.
we are wholeheartedly thankful to all the resource persons, those who spared their valuable time and share their huge experience regarding of this entire subject of this plant sciences. So our fruits shown like Shivaji Naik. So
This is
many.
all about your guidance and subject awareness. This is from that, sir.
Really, my dear Shivaji Naik, Rambabu sir has huge experience. Personality is my beloved brother. Okay,
Yes, sir.
not
Yes, sir.
only Rambabu sir is not a single person. He's an energetic and entire the captain of the entire team of the Misal, entire team of the Oasis team, entire team of the, even though the faculty members, those who are going to train as a resource person of in this our honorable commissioner sir has selected as a resource person, and also is an also coordinator of the training classes already done in training program has done in Warangal region recently. Sir has acted as a entire training program coordinator and successfully more than 10 days, more than 12 days, and before the training classes, more than 1 month, he has given a wonderful time for the arrangement of the all type of the arrangements for the successful completion of the training program. So because of his hectic schedule, sir is also now in the NSS camp also in Kakati University. But regarding of these classes, sir has given this wonderful time. Really, we are wholeheartedly thankful to you, my beloved brother, Dr.M.Rambabu garu. Okay, your class is.
Any doubt? Any
Your
doubts,
class
my dear?
is energetic and very fantastic. Am I right, Shivaji and
Thank
Prakash?
Yes, sir. Yes, sir. Absolutely.
Right. Really, hats up to you, sir.
I will feel very happy whenever ask a single question. My students ask a single question that shows more happiness to me.
Yes, sir.
Ask any question.
Shivaji.
Regarding
Prakash.
this class.
Sir, you just tell about protoplast culture, sir.
Okay, protoplast culture. I will take the tomorrow class is protoplast culture. Anyhow, protoplast are the cell wall-less cells.
Yes, sir. Yes, sir. Cell without cell wall that could be cultured
Cultured
under
on
aseptic
the
medium.
But the one thing is, after the culture of that protoplast, that cultured protoplast must be generated. First of all, we are removing the wall by the using of enzyme mechanical method. After the culturing that protoplast, in the culture room, that protoplast must be generate the wall, converted into cell. Then after only it will be going to regenerate, it will go into callus formation. Then further process will be carried. I will take that class, protoplast culture, tomorrow.
Okay, sir. Okay, sir.
I'm
Yes,
happy
sir.
now.
Sir, share this class in group now.
Yes,
Sure.
definitely. Definitely, my dear Prakash. Our beloved Guru, Dr.M.Rambabu sir is always with us, my dear Prakash. Your
Yes, sir.
enthusiasm
I think
really
Prakash
acceptable and invitable and also appreciable, my dear Prakash.
And
Prakash
by
is, I
your
think, our
yes, sir.
KDC student.
Yeah, sir.
Okay.
So really, Prakash is very
Sir,
dynamic
sir,
personality.
one question, sir.
Right.
Sir.
Happened.
Sir, hello.
Happened.
Sir, why we are performing this tissue culture culture, sir. Apart from the general yes, this conventional method, why we are doing?
Actually, each and every technology is helpful for the crop improvement. For example, this question is moves to the application of plant tissue culture. Whenever we are going to see the application of plant tissue cultures, you get the answer. Mass multiplication will take place from this tissue culture. For example, in the single seed, you are going to sow a single seed in the soil. What happens automatically? A single plant will be developed. But here, you use a single X
We
plant.
generate multiple plants, sir. Yes, sir. Yes, sir.
Multiple plants. The
Yes, sir.
time gap is also very low.
Yes, sir. Lifespan is very low.
Lifespan, not lifespan. The production
Yes,
time.
sir. Yes, sir.
The
Yes, sir.
production time is also very low.
Yes, sir.
Low
Yes, sir.
time consumption, high amount of plant production. That is the
Yes,
quality
sir.
of tissue culture.
Okay. Okay.
Are you happy? Satisfied?
Yes, sir.
Yes, sir. Yes, sir.
Okay, I will take the class tomorrow also. Don't worry, my dears.
Okay, really, thank you very much, sir.
Okay, sir.
Tomorrow, same time, we will meet.
Right, sure. Thank you.
Okay, thank you.
Thank you, sir.